My name is Jouni Tuisku, and I'm a postdoctoral researcher in Turku Pet Center, where I'm working with the pet modeling things. And at this meeting, I'm presenting a framework that we have developed for the automated processing of total body PET CT data, which aims to improve the efficiency and accuracy in the data analysis. So why do we need the automated processing? Well, here you can see a typical radio weather PET image that has been scanned using our total body PET CT scanner. And as you can see, the large field of view of the total body image brings an increased amount of data that needs to be processed. But unfortunately, the manual processing is very tedious and time-consuming task. For example, studying the interaction between the different body regions requires drawing many regions of interest. And for just one subject, this could take even several days, of course, depending on the number of organs studied, which is not very practical. And this highlights the clear need for automated data processing to make the workflow faster and more efficient. So considering this, we have created our own method for total body PET data analysis. And this flow chart here shows the main components of the process. And our method is called the Turku body pipeline, or shortly Turbo. And it is divided into two branches. And one is for brain image processing, and another is for the other body organs. And first, the brain is extracted from the total body image using our own automatic software, and then the brain CT image is aligned with the brain PET image. And following that, our existing Magia pipeline is used for the brain PET data analysis. And next, the other body organs are processed, and it starts with segmenting the CT image into several different anatomical structures. And then the PET image is corrected for subject motion, after which the CT and all the segments are registered to the PET mean image. And finally, the PET kinetic models are applied for regional time activity curves, and as well as for the voxel level data, after which all the results are saved in a database for later data analysis. And currently, this method is applicable for radio water and FDG PET studies, but it can be extended later to cover other radioligands if needed. So because the brain PET processing is quite straightforward, I'll talk next more about the different total body processing steps, and then I'll show how we have validated our pipeline. So let's take a closer look to the quantification of the body organs. And here you can see the CT image that is currently segmented with a tool called Total Segmentator. And this tool produces regions for about 100 different anatomical structures by using machine learning methods. And this figure shows the segmented regions in more detail. And this tool segments all the major organs and the skeleton and muscles and cardiovascular structures, such as the aorta and left and right ventricles and the portal vein. And even with our low-dose CT images, the quality of the segmented regions it's very good, although they are still relatively large entities. And especially these segmented cardiovascular structures allow for creating an automated input function for quantifying the PET data. And for the modeling, we have chosen to use an image-derived input function for the descending part of the aorta, which works well for analyzing most body organs. And the input is extracted using the same criteria which are used for manual drawing by our own imaging experts. And here in these figures, you can see example cases where the automatically derived region is shown in white color, and the manually drawn ROI is shown in blue color. And there's quite good overlap between these two regions. It is also possible that there are mismatches between the CT and the PET images that would need to be corrected. And ideally, these misalignments would be corrected already during reconstruction. But however, no suitable methods are currently available for this purpose. And therefore, the misalignments need to be corrected using post-reconstruction methods. And here we have chosen to align the CT image to the PET, which then ensures that the PET measurement is PET measurements would remain unaltered. And in order to get a good match, the CT is sort of elastically co-registered, which also improves the alignment of the CT-based ROIs with the PET image. The second issue is the subject movement during the scan. Even if the subject would lie still, the heart is still beating and the subject is breathing, and this will have impact to also to the nearby organs. And here in the left figure, you can see a typical case showing the heart movement. And to address this, we can apply the basic rigid object transformations like rotations and translations for correcting the movement. And these operations are applied so that each frame would match the reference frame. And because the rigid transformation treats the image as a fixed object, it doesn't allow any stretching or shrinking, but for heart and aorta, this works reasonably well, and then we can get a motion corrected input function for kinetic modeling. It's also possible to apply the motion correction for the total body data, but here obviously the rigid correction methods are not sufficient. And here in this video, you can see, for example, that the breathing and heart motion are corrected relatively well in the right video, and the resulting image seems to have some motion, but it's not so severe anymore. And here we have applied a so-called diffeomorphic method, which extends the rigid transformations by allowing also shearing and scaling, and then further fine-tuning is obtained using nonlinear transformations. So after all these corrections, the time activity curves can be extracted from the PET image, and then the modeling is carried out for each organ and at both regional and in voxel level. And for both radioligands, the standard quantification methods are implemented in this toolbox. And as the pipeline is modular, we can add different models or settings for different regions easily. And as a result, the plots showing the model fits and the used input function are produced for each region, where the model quality and the fit accuracy can be easily checked. And the resulting slice plots showing the model, showing the voxel level parametric images are also saved for quality control. And additionally, the regional parameter estimates can be checked individually and collected for the chosen list of subjects for later statistical analysis. And to quickly check the results of co-registration and motion correction, we have included also quality control plots where the segmented regions are first overlaid with the CT and then with the PET mean image. And the motion correction results can be evaluated in every segmented region separately So for example, we can observe how the movement of a single voxel at one segment border is corrected relative to the chosen reference frame. And for both of these radioligands, the reference frame is selected as the one that has the most similarity compared to the CT image. So we have tried to simplify the use as much as we can. And the PET and the CT DICOM data need to be stored to separate folders, after which the processing is started with a simple command. And in our PET center, the processing is integrated with our own IBO database, and all the required information is read from the image headers and then saved in our database for later use. But the pipeline can be used similarly without the database connection, so that's not required. The total running time is approximately 1 to 2 hours, depending on the number of frames and number of available CPU cores, and if the GPU can be used for tissue segmentation. But it is relatively fast compared to the amount of the data. The processed files and the results are stored in the separate folders, which also contain the log files and quality control plots and options. And the motion corrected data is located in the subfolders within the anatomical and the PET data folders. So that was all about the method description, and now I'll move on to show how we have validated the automated method. And we have evaluated the processing using 2 different data sets. The first set includes radio water data, which contains 21 older control subjects from our larger coronary artery disease study. And here the subjects were scanned in 2 conditions, at rest and during adenosine injection. And the second dataset contains in total 16 lean and obese subjects which were scanned with FDG radioligand. And for both datasets, our experts manually drew ROIs for input and target organs, which were then used as a benchmarks to assess the performance of the automated processing. And based on the visual inspection, the one tissue compartmental model fits of the radio water data, and then the padlock model fits to the FDG data. They both have good quality in general, but some regions for radio water data, such as pancreas and kidney, might require some additional modeling steps to improve the fits. But overall, the fits look very good. The correlation between the modeling results of manual and automated input function is actually nearly perfect in radio water data, and moreover, the Bland-Altman plots show no significant differences between the manual and automated inputs, which demonstrates that the automated approach is very similar to the manual approach. And there's also strong correspondence between the manual and automatical inputs in brain and muscles and in heart in FDG data. But the correlation is slightly lower in regions with a lower metabolic rate, such as in pancreas and in spleen and in liver. But overall, there's a good agreement between the 2 methods. And the regional parameter estimates also show strong correlation with the voxel level regional averages for both radioligands. But however, in the radio water high flow regions, the voxel level estimates are slightly biased because of the coupling of the arterial volume fraction and the K1 parameter that describes the rate of blood flow. And for this reason, the arterial volume fraction needs to be fixed in the voxel level estimation. And for instance, in kidneys and in heart myocardium, there's more bias as compared to the other regions, which with the lower blood flow. So in summary, our automated image processing and modeling toolbox is highly efficient in analyzing data from multiple organs with minimum manual labor. And overall, this approach is much more faster compared to the manual processing, and it creates reliable and reproducible results. And all the tools that we have used are included in our pipeline are free for academic use, including the in-house software we use at Turku Pet Center. And the final software is intended to be fully automated and to work with just image files as inputs, so no previous knowledge in programming is needed. And currently we're working on a manuscript about this pipeline, which should be ready later this year. And after this, the code will be available for everyone. And now I want to thank the other project members for their contributions, and also thank you for your attention.